So after this extensive talk by Dr. Rajiv, I am going to cover the liver regeneration, particularly in focus with the liver injury, where I will basically cover the regeneration in normal liver turnover, what is the role of stem cells and the hepatocyte, regeneration with respect to partial hepectomy, what is the mechanism starting from the initiation, progression, and termination of the regeneration, and the role of macrophage in PHX induced regeneration. So how it is acting as a master regulator. The next we will describe about the regeneration in liver injury in details, the mechanism for chronic, acute, and acute on chronic, what are the limiting step, and the hepatic stem cells, or whether it is a hepatocyte. And lastly, I will talk about the bone marrow response and the gut-derived barrier dysfunction in regulating the limiting step of regeneration. So coming to the regeneration in response to normal turnover, this is required because though we see hardly 0.2% hepatocytes in the proliferative stage in normal liver, but this is also very critical for maintaining the normal homeostasis. So in the initial studies using the proliferating dye, the people have come to the conclusion that in the liver, particularly in the canal of Hering area, there is a cell population which we know that hepatic stem cells, and the central vein area is the terminal differentiated hepatocytes. And what they propose that this is a streamline hypothesis from where the stem cell concept came up, that there is the aging of once these terminally differentiated hepatocytes aged and die, the cells from the canal of Hering area is basically the one who is basically replenishing this aged hepatocyte. Later in 2015, there is another concept came which basically object the canal of Hering as the only site of hepatic stem cells. So what they say is that even in the central vein area, surrounding the central veins, you have, though these cells looks like hepatocytes, but they have the stem cell-like characteristics, and they can self-renew, and they are the one who is basically participate in the normal homeostasis or maintaining the dying cell repopulation. But later, there are various studies came up with the lineage tracing experiment, which disprove this hypothesis of a stem cell as a primary factor which is leading to the normal turnover of the liver. In case of normal homeostasis and injury, using the lineage tracing of the stem cells as well as the biliary cells, they clearly proven that they are not participating in the normal turnover. Other study which in 2018 came, which is more sophisticated in terms of they label the TERT, and what they shows that throughout the liver there is a population of hepatocyte which is having a high TERT expression, and they are the one who is basically take part in both homeostasis as well as in case of liver injury. After that, this concept start emerging and using not a lineage-based tracing, but rather than using in an adult liver, they label the different region of the hepatocytes, and then they concluded that it's not the stem cell population, rather than all the hepatocytes present in the liver, they have a capacity to repopulate. In 2020, this annexin 2 positive hepatocyte was disproven, and it was shown that this annexin 2 or LGR5, which they are claiming, they are facultative in nature. So once the hepatocyte start proliferating, they start acquiring these type of markers, and that could be the reason that there is a false interpretation of these cells as hepatic stem cells. Apart from this lineage tracing experiment, initially also it's a very good established data that when we use the serial transplantation for hepatocyte repopulation, so it also says that hepatocyte has a tremendous replicative capacity. So even this, and there is no demonstrated loss of hepatocyte or its regenerative capacity. So what all this study concluded that there is no role of hepatic stem cells in normal turnover. There is equal participation to all hepatocytes in democratic manner. That means if the cells are losing in the portal area or a central area or a mid-zone area, the cells, hepatocytes which is near to that dying or senescent cells, they are the one who is basically proliferate and replenish those cells. Next, which is the liver regeneration in terms of partial hepectomy, which is well studied in both human as well as in animal model. So most peculiar thing about this regenerative model is there's no or minimal injury to the liver. Remaining liver environment is usually sterile, and the studies using animal models has clearly shown that there is a multiple regenerative mechanism is occurring during this in case of partial hepectomy, both conventional as well as in non-conventional manner. So in non-conventional manner, if the hepectomy is less than 30%, usually they take the non-conventional way. That means either there is a hepatocyte hypertrophy, or the binucleated hepatocytes simply go to the cytokinesis and make the two new hepatocytes. That is the major way of hepatocyte regeneration. However, when the hepectomy exceed beyond the 30%, then the hepatocyte proliferation is the major way of hepatic regeneration. And that occurs in a very step-wise manner and tightly regulated also. And this clearly so regeneration occurs through the sequential proliferation of mature hepatocytes and non-parenchymal cells. In this case of injury, some non-parenchymal cells like endothelial cells and macrophages are also get contributed by the bone marrow derived cells. Hepatocyte proliferation occurring as a gradual wave from periportal to central area, and this is very critical because we will discuss the later, but when the hepatocyte start proliferating, they loses its differentiation capacity. So to prevent the liver failure, that if all hepatocytes started proliferating, that can lead to the liver failure. So that is why the nature has adopted this mechanism that regeneration doesn't occur in response to partial hepectomy in throughout the liver. So starting with the very first step, what is the state of liver in case of the static stage? When the normal liver, there is no shear stress in the liver, continuous supply of TGF-beta by the L6, which act as a brake to the hepatocytes. The hepatocyte HGF as a growth factor, it is present, but it is bound to ECM and not available. Apart from that, also the emerging recently shows that ECM hepatocyte interaction, particularly this kinase, integrin-linked kinase, also provide the brake to the hepatocyte proliferation. So just after the partial hepectomy, once there is an increase in portal flow, that lead to the shear stress in the endothelial cells, and within less than 15 minutes, you will see the change in the pro-HGF urokinase activity is increased, that lead to the from pro-HGF to HGF. There is a destabilization of the endothelium that leads to the loss of the TGF-beta signaling, and this way there is a break, whatever the brake was there in the static liver, normal liver, that brake has been removed, and this lead to the migration of Notch and beta-catenin signaling. MET and EGF receptors get activated, and that also lead to the expression of various stem cell associated proteins like alpha fetoproteins and all this. So that basically trigger the regenerative response in the very early stage. Later on, this is the most important and rate limiting step in hepatocyte proliferation, and which is basically catered by the Kupffer cells. So apart from increasing the shear stress, the endothelial, this increased portal flow also lead to there is an increase in the gut factors like complement LPS, and it also lead to the increase in the vagus signaling. All this signaling in the Kupffer cells lead to increase in the this IL-6 production or TNF-alpha production by these cells, leading to the activation of the IL-6 STAT pathway, which is a very critical component in priming the hepatocyte to enter into the regenerative phase. So what it basically does, it increases the transcription and translation of the cyclin D1, which is required for entering the cells into the S phase cycles. Together with the other mitogenic factors, it also helps in the metabolic adaptation, particularly the transient steatosis, which is very critical component, and it also leads to downregulation of hepatocyte differentiation marker and loss of function. So this way, now the hepatocyte are ready to enter into the proliferation, and then there is a cascade of events or the growth factors has been produced at the different level. Like in the beginning, the HGF, which is a more prominent growth factor for the hepatocyte proliferation, it is given by the activation of the hepatic stem cells, and later on it is mainly given by the LSEC. So Once the hepatocytes start proliferating, these proliferating hepatocytes also secrete various growth factors that help in the proliferation of the other cells like endothelial cells, macrophages, cholangiocytes like HGF, IL-6, and TRG signaling help in the cholangiocyte proliferation, VEGF, NGA, TGF, FGF, they help in the LSEC proliferation, and the GM-CSF and MCSF from the HSC that regulate the Kupffer cell proliferation. And in the animal model, it is usually shown that by the day of 7, all this hepatocyte proliferation has basically slowed down, and most of the cells following the hepatocyte proliferation, they also start declining. In termination of the liver regeneration, initial belief was that it is the TGF beta which is the major player of that they start regain, and that is the cause of the failure of, the termination of the liver regeneration. But the newest study, what this is that ECM hepatocyte interaction through the ILK is one of the master regulator of normal liver size and is involved in the restoration of liver weight to the pre-partial hepectomy levels. In normal mouse liver regeneration, the expression of ILK is enhanced, and the most interesting thing is that when they knock down this signaling, what they found that even after the completion of the regeneration, the total gain of the mass was exceed, that is from 100 to 158%. So that is the reason they now start focusing on the ILK mediated signaling as one of the major cause of the regeneration termination. TGF beta inhibits hepatocyte proliferation in the culture media. However, no clear association of TGF beta and termination of liver regeneration has been established till date in case of in vivo experiment. Interestingly, recently this paper came which says that though we think that hepatocyte regeneration has completed by the day of 7, what they shows that even though all the cell proliferation has been halted or it's over, but if you look at the vascularization is not completed even at the day of 14. And what they shows that this initial increase in the shear stress that also lead to the senescence in the endothelial cell population. And with this senescence cells has to be removed rapidly to complete the regeneration, particularly the vascularization in this sinusoidal area. And there they says that with this senescence, there is a rapid infiltration of the monocyte from the bone marrow, which differentiate into the macrophage, and they are the one who is basically helping in the clearing of this senescent endothelial cells. And once this is completed, then the complete recovery of the lost liver mass is achieved. So in summary, if we talk about the regeneration in PHX, no or minimal cell injury, regeneration is clean and begins without inflammation. Regeneration, self-replication of a mature hepatocyte followed by the other non-parenchymal cells. In the early phase, contribution of the bone marrow is limited. However, in the late phase, we can say that bone marrow monocyte-derived macrophage are critical for clearance of the senescent L6. Both primary mitogens are critical, like EGF and HGF, both are critical for this one. Source of HGF, ECM, and HSC in the early stage and LSEC in the later stage. Source of EGF is the Burner glands of the duodenum, HBHGF from the Kupffer cells, and the HGF from the LSEC and Kupffer cells, and ILK signaling is the one who is basically stopping the regeneration process. So if we consider the entire regenerative response, what we found is that liver macrophage act as a key player. First, it is the one who is helping in the hepatocyte priming step, which is the rate limiting step in this process, and also in case of vascular maturation. And then also the study says that depletion of KC impaired the function or delay the liver regeneration process. So we can say that macrophage has some important role even in case of partial hepectomy, initiated liver regeneration. So how the regeneration fail whenever you have extended hepectomy, and what are the mechanisms which has been shown is that there is excessive portal flow or that provokes the sinusoidal endothelial cell injury. That could be one of the reason. Also, there is a secondary to portal hypertension. There is a increased gut barrier dysfunction and predisposition to the bacterial translocation that can also hamper the regeneration. But nutshell is that there is a dysfunction of retroendothelial system in case of extended hepectomy, which has been shown as a major cause of regeneration failure in this kind of model. Next is the liver regeneration in case of injury, like ischemic, toxic, or infectious type of liver injury. Unlike PHX, there is a necrosis and heavy inflammation. Active contribution of the bone marrow is obvious, and regeneration begins with the removal of dead crops and subsiding the inflammation. So first thing is that what cell participate in the regeneration? Very beginning, it had been shown that even in case of acute liver injury, the robust hepatocyte proliferation is the primary way of hepatocyte regeneration, and that too is a dose-dependent response, so that the more the injury, there is a more hepatocyte proliferation, and that ratio occurs to the certain extent of injury. And after that, though the injury proceeds, there is a failure of regeneration. Recently, also using this more sophisticated tools like transcriptional method as well as the in vivo cell imaging system, what they come to the conclusion that there is a robust hepatocyte proliferation. Incoming pericentral hepatocytes changes its morphology, elevate protein turnover, and upregulate the oncofetal genes, which is again saying that there are some de-differentiation going on during this regeneration process, and there is a down-regulation of MHC1 and MHC2 in the Kupffer cells to protect the autoimmunity. In case of acute liver injury, yes, there are 3 major studies which has concluded that there is a role of hepatic stem cells when the hepatocyte proliferation is not working. And what they saw, the 2 studies saying that this is coming from the canal of Hering area, and another study which says that there is a hybrid hepatocyte in the periportal area. They are the one who is participating in this process. However, this was again challenged by these lineage tracing experiment, which clearly says that there is no role of the hepatic stem cell in case of acute liver injury, induced hepatocyte regeneration also. And particularly this last study clearly demonstrates that whether it is a pericentral or periportal injury. So if you have a periportal injury, the hepatocyte from the pericentral area is participating in the repopulation, and if it is a periportal, it is the other way around. And the second interesting point in this study, what they shows that when the hepatocyte start proliferating, they start acquiring the marker of stem cells like anaxin or LGR5. So what they concluded is that this stem cells, what we observe in the hepatocyte, in the liver, they are facultative in nature rather than a true stem cell population. So how they are coming up, this study also shows that just in the margin, where you have the necrotic margin, the hepatocytes are changing its morphology and they are acquiring the expression of various oncofetal protein. The another study also shows that the source of this progenitor or ductal cells are basically the hepatocyte, which is leading to de-differentiation of hepatocytes is the major source of oval cells, or what we call as the hepatic stem cells. Regarding growth factors in case of acute liver injury, unlike HGF and EGF, which were both critical for the partial hepectomy, the most of the study suggests that the EGFR or EGF signaling is the most critical in case of acute liver injury. However, unlike PHX, they have a biphasic response. So in the early phase, the EGFR signaling increases the mitochondrial damage and can lead to the hepatocyte injury. But in the late phase, EGFR activation are indispensable for hepatocyte proliferation and regeneration. So the question is that why after certain extent of injury or the acute liver injury, there is a failure of hepatocyte proliferation. So this was extensively studied by the Bhusan et al. And what they have used in this elegant experiment, they use the doses, the 300 and the 600. So 300 you have a active regeneration, and 600 there is a regeneration failure. So what they say is that in the higher dose that they lead to regeneration failure, though the hepatic mass, the remaining hepatocyte mass is more than 50%, but there is a delay in regeneration. And there is a delay in clearance of this necrotic area. Further study, analyzing the different growth factors and signaling, what they conclude is that like all the important mitogenic factors like EGF, HGF signaling are still very high. However, they found there is a defect in the TNF alpha and cyclin D1 signaling, which is mainly catered by the Kupffer cells, and particularly in case of delayed clearance or the induction of the hepatic regenerative response. Later, the Stewart group shows that this necrotic area, particularly in the lining of the remaining hepatocyte, there is a rapid accumulation of the bone marrow derived macrophages, and they are the one who is basically producing the TGF-beta and causes the senescent like appearance in the boundary area. There is a recently published paper which also favor this kind of finding, and this says that the macrophages produced by the TGF-beta, macrophage given TGF-beta is basically responsible for the transient senescence in this perinecrotic area. But question is that whether this transient, apart from that, the study also shows that there is a change in this morphology and acquiring the stem cell-like property. So whether it is helping or whether it is inhibiting the regeneration process, so this study has clearly shown that this acquired property of staminase or this transient replication senescence, in fact, is a protecting mechanism for this propagation of the injury from, so that basically prevent the rapid progression of the liver, so hepatocyte injury. So what they say is that this macrophage induce changes in the hepatocyte, like they start expressing the SOX9, the stem cell marker, and these hepatocytes, when they express the stem cell marker, they become more resilient to the injury and they are more protective. So this way, they are creating a kind of wall surrounding this necrotic area to prevent the further damage of the remaining hepatocytes. This is also proven by the other studies also that the macrophage induced clearance of this necrotic area is very critical for initiating the regeneration process. Like in this study, what they shows that if the macrophage lack the MERTK, which is required for the neutrophil clearance, they are defective in regeneration. Recently, there is other paper came that apart from this clearance, they also says that once this macrophage gets activated, there is an increased recruitment of the eosinophil, and that eosinophil help in the increased production of the HbEGF by the Kupffer cells that help in the production of the growth factor for the hepatocyte regeneration. Not only this, the experiment using the exogenous transplantation in ALF injury clearly shows that in normal ALF acute liver injury, the native liver macrophage show defect in the clearance of the dying cells. However, if you infuse a healthy macrophage into this, that lead to increased clearance of dying cells as well as promote the hepatocyte proliferation. So in summary, if we say the acute liver injury and the regenerative response, so unlike the partial hepectomy, just after the initiating signals, the hepatocyte enter into the proliferation phase. In case of liver injury, you have a massive necrotic area which has to be cleared before the initiation of the hepatocyte proliferation, and it acts as a rate limiting step. So, and during this process, the bone marrow plays an active role. Coming to the chronic liver injury, in case of chronic liver injury, there are various mechanisms has been shown that could act as a potential factor, like excessive scarring, which can inhibit the regenerative response. There is a replicative senescence of the hepatocytes. That means hepatocytes loses its proliferation capacity, and also there is a defective macrophage clearance of dying cells and efferocytosis dysfunction. So we will go through and see that which is basically the major player. So coming to the hepatocyte proliferation per se, yes, the clinical study also shows that. So with the increase in liver injury, if okay, quickly. So in case of chronic liver injury, if we talk about the hepatocyte proliferation, we'll discontinue. Come to the last thing, saying that what basically. So summary, if we talk about this, we have already seen that this is the rate limiting step where macrophage plays a major role. Bone marrow response is critical for doing all this process. So first we try to see, this is ILBS data from Dipanshu, which is not published yet. So how the bone marrow response change with respect to liver injury. So if we see the injury per se, it is peak at 6 hours, then 24 hours by 72 hours. However, the regenerative response occurs starting in the recovery phase. So that again says that the clearance of injury is very critical. If you see this see the bone marrow response in this case, what we observe that this active myelopoiesis or increased production of myeloid cell is also happening at the recovery phase. And at the peak injury phase, there is a reduction in the LTHSC, which is basically and followed by the rapid proliferation of the myeloid lineage cells. And that too, so if we also look, because biogenic adaptation is required for the transiting this hematopoietic stem cells from the stemness to the, and what we see found is that biologic adaptation precedes cellular recovery. Later in the chronic injury model, the Nidhi, which is our, again, a PhD student, he clearly shows that the importance of bone marrow, particularly in chronic liver injury. So what she gives the conclusion that during the chronic liver injury, till the stage of compensated cirrhosis, you have an increase in fibrosis and you have an increase in regeneration also. But from transition to compensated to decompensation, though the fibrosis remain unchanged, not much difference, but there is a rapid fall in the regeneration. And the most important finding of her thesis was that before the beginning of this, there is a loss of hematopoietic stem cell reserve, and that reserve is leading to as the major cause of the loss of the macrophage number and function in case of chronic liver injury, and that lead to the transition from the regenerative phase to the regeneration failure during the course of chronic liver injury. She also showed the proof of concept that if you can rejuvenate the bone marrow, you can prevent this process and you can delay the transition from compensated to decompensated cirrhosis. Later, more in-depth in the mechanistic way that Dipanshu has taken as a PhD challenge, and what he recently shows that, so this I would like to emphasize that what he shows that when in the liver injury process, so you have the damage of gut also, you have the damage of liver also. So when there is both Kupffer cells is damaged as well as the gut is damaged, that too leads to the very rapid increase in the endotoxemia in the bone marrow. That endotoxemia, though it causes the increased myelopoiesis, but what happens is that this production of the myeloid cells, if we look at, they are giving a very peculiar type of myeloid lineage cells. They are defective in the macrophage, and what they show is that this increased TLR response, decreased glycolysis, mitochondrial aspiration is though high, but there is a mitochondrial defect, oxidative stress is high, and there is a bioenergetic dysfunction. And when these cells are allowed to differentiate, they fail to differentiate into the mature macrophages. And once they reaches to the liver, they are more of inflammatory rather than be able to do the clarification of the dead crops. Home message is that robust hepatocyte proliferation accounts for liver regeneration in both PHX as well as in liver injury. Hepatic stem cells are facultative in nature, either they are inadequate or not participating in the regeneration process. Hepatocyte priming in a PHX and clearance of apoptic necrotic cells as a rate limiting step. Liver macrophages act as the key to liver regeneration. Why I say that? The primary hepatocyte for regeneration and clearance of the senescent cells, clearance of the apoptotic and necrotic cells, hepatocyte dedifferentiation, transient replication arrest, provide growth factor for regeneration, and bone marrow homeostasis maintain the increased requirement of mature functional liver macrophage pool. Gut disruption can decrease the production of mature functional macrophage needed for this process. So my way forward is that even though we think that regenerative therapy as a potential to transplant-free management of this patient, but if we look at the clinical study, still whatever the therapy has been used, like HSC, MSC, or bone marrow macrophage, they are still showing the very transient effect. And that is the reason I propose that we should, rather than having only a regenerative approach, we should have a stepwise and multifaceted layered approach to prolong the regenerative effect. Like, we know that gut is the source of injury and the bone marrow is the source of repair and regeneration. We have given you the data that gut can damage this also, gut can damage this also. So the strategies, like together with this therapy, like you also have a bone marrow regeneration, as well as the therapy like FMT or therapy to prevent or restore the gut epithelial barrier, should be considered to make the regenerative therapy more successful. Thank you.